Now, finally, we can discuss the last of the signaling functions of the lipid molecule, specifically those of the fatty acids in the form of second messengers and eicosanoids, a group of phospholipid metabolic derivatives that have very important regulatory autocrine and paracrine functions in the body, regulating the cell cycle, but also various inflammatory processes. Let's get started. An important reminder about some of our unique dietary fatty acids. So remember, we can synthesize saturated fatty acids and monounsaturated fatty acids, but polyunsaturated fatty acids like omega-3 and omega-6 fatty acids cannot be created de novo. We simply do not have the delta-12 and delta-15 desaturases required, and instead we have to get them from our diet. Really, it's hard to avoid trace amounts of omega-3 and 6 fatty acids in our diet, so most people are not deficient, as we'll discuss. But of course, calling anything essential in the diet has a different scientific and medical connotation than it has to the public, who then fear that diets low in these are necessarily dangerous. As we've discussed, we know the value of MUFAs and PUFAs in stimulating our exogenous pathway of fat anabolism and actually blunting the potentially dangerous endogenous pathway of fat anabolism, which involves increasing triglycerides and VLDL, which is of course a certainly attractive mechanism by which we can favorably manipulate our fat intake in our diet to optimize our nutritional intake for health reasons. But let's see if there's anything else to these fatty acids that's worth considering. Specifically, we keep saying omega-3 and 6 fatty acids, but let's get a bit more granular. Omega-6 fatty acids come primarily in the form of two acids, linoleic acid and arachidonic acid. Arachidonic acid is conditionally essential, meaning that if you get enough linoleic acid from vegetable oils, you can make arachidonic acid on your own. But eating meat, poultry, and eggs, you can just get arachidonic acid directly in your diet. With these dietary sources, the combination of vegetable oil and meat and eggs, it's pretty hard to miss omega-6 fatty acids. And indeed, some epidemiological studies of Western diets have pointed towards an abundance of omega-6 fatty acids, especially compared to omega-3 fatty acids. But what about omega-3 fatty acids? We are now talking really about three specific fats. Alpha-linoleic acid, found in vegetables, seeds, and nuts, which can be metabolized into two important omega-3 fatty acids only found in fish and fish oils in eicosapentaenoic acid, or EPA, and DHA, or docosahexaenoic acid. Both EPA and DHA are therefore actually conditionally essential as well, like arachidonic acid. What is interesting from a bird's eye view in the epidemiology is those people who have higher diets consuming DHA in particular find a lot of it in the cell membranes of the central nervous system, and thus those low in DHA find less DHA there proportionally. This has been used as a partial basis hypothesis for differences in Alzheimer's disease predilection amongst patients who are ApoE4 variant carriers, as an example. Overall, we shuttle all of our omega-3s and 6s that don't immediately get stripped for parts in beta oxidation to the plasma membrane of our cells, where they sit and reside as plasma membrane constituents. Importantly, they don't just sit there idly, or at least the hypothesis is that the membrane composition of our cells isn't just a reservoir for fatty acids, but also participates in direct cell communication and signaling and can actually affect cell function. Further, these specific omega-3 and 6 fatty acids, and only these, as we'll see, can be metabolized into an important group of molecules called eicosanoids. But briefly, to answer the question that we're all asking, can we actually be clinically deficient in omega-3 and 6 fatty acids? And the answer is yes, but no. Essential fatty acid deficiency, or EFAD, is an extremely rare nutritional deficiency, really isolated only to neonates with inborn errors of metabolism. The reason why it's so rare, despite the hype on the internet about insufficient omega-3 and 6 fatty acid intake, is the dietary redundancy of these fatty acids. As we've seen, even if you don't eat oily fish, you can still get omega-3s, at least the minimum required for homeostasis, through vegetables and nuts or soy or canola oil. Diets low in meat, poultry, and eggs for omega-6s can be compensated for diets that have vegetable oil, which is very cheap, or cow's milk, which is ubiquitous. This isn't to say getting the minimum amount of these fats is optimal, but it certainly isn't immediately life-threatening and causing EFAT. Deficiency syndromes have, however, shown us the consequences if these patients that do become deficient develop intellectual disability, clearly playing a part with scaly skin, alopecia, and even thrombocytopenia as consequences. But I said we need these fats to create eicosanoids. But what are eicosanoids? These are autocrine, paracrine, or even endocrine tissue hormones that are derived from metabolizing arachidonic acid or other 20-carbon PUFAs from the Greek eikos, which means 20, so that includes eicosapentaenoic acid or EPA, the omega-3 fatty acid. Eicosanoids serve various functions which vary from cell growth and the cell cycle to physiologic action required in pregnancy and even the management of inflammation. These molecules continue to be studied, but we know a fair bit about three groups of eicosanoids which you have likely heard about: the prostaglandins, the leukotrienes, and the thomboxanes. All of these require some 20-carbon PUFA precursor, and when these are pulled from our plasma membrane, we can synthesize eicosanoids for important cell signaling roles. Less understood eicosanoids include molecules called resolvins, lipoxins, and aoxins, which are very poorly understood but interesting basic science. One question that has been asked is there a difference between the eicosanoids you make if your body pulls out arachidonic acid or if it pulls out EPA? What about meat acid and omega-9 fatty acid? The answer to that appears to be yes, and some hypotheses have already been formulated about the consequences. For instance, you can see that obtaining eicosanoids from omega-6 fatty acids from meat and vegetable oils may favor production of 2 and 4 series prostaglandins and leukotrienes. So for example, PGE2 and PGE4 versus with EPA probably favoring 3 series, aka PGE3 and PGI3. Omega-9 fatty acids may be able to make no prostaglandins or thromboxanes. You can see some other similar differences in the leukotrienes produced, but interestingly, we can de novo synthesize omega-9 fatty acids from oleic acid, which is of course in olive oil, but we'd still expect to be able to make prostaglandins through trace dietary intake of precursors to omega-3s and 6s. The cell pulls either arachidonic acid or eicosapentaenoic acid from the plasma membrane using an enzyme called phospholipase A2, which starts the pathway of metabolism into these eicosanoids. Importantly, however, if both EPA and arachidonic acid pathways are simultaneously occurring, consider that they might compete for enzyme activation and auto-regulate each other, i.e., it's not obvious that these two pathways are mutually exclusive. The first class of eicosanoids is prostaglandins, which are extremely important molecules in every cell in all animals. They are named conventionally as P for prostaglandin, followed by a letter based on their ring structure, and then a number based on their number of double bonds. They were initially found in the prostate gland and thus named, but they are in fact ubiquitous in the body. See on the pathway to the left. We use PLA2 to liberate arachidonic acid or EPA from the membrane, and then use an important enzyme called cyclooxygenase or COX to metabolize the fatty acid into a group of precursors that eventually becomes one of the lineages of prostaglandins based on further enzymatic metabolism. An important group of drugs, including the over-the-counter ibuprofen and acetylsalicylic acid, inhibit this cyclooxygenase enzyme on purpose to prevent eicosanoid and thromboxane production. This is because some prostaglandins are associated with pain, and reducing the production of these has been thought to blunt this response. Depending on the prostaglandin, there may be very wide varying effects. Just take the prostaglandin GE1 or PGE1, which is a vasodilator, but also reduces gastric acid secretion. We have two drugs that mimic PGE1. The first is alprostadil, which we use in erectile dysfunction, but also in babies with patent ductus arteriosus, since it's used to dilate arteries, and in whom we'd want to maintain the patency of that ductus. NSAIDs, of course, on the other hand, like ibuprofen, would close the ductus, since they'd stop us from making PGE1. Since vasodilation may be useful to lead to more peripheral blood flow, like I said, PGE1 analogs can be used in erectile dysfunction. Misoprostol is another PGE1 analog that can be used for multiple things. First, to protect the stomach from acid erosion when given with NSAIDs, but also as an adjunct to medical abortion, since it ripens the cervix and can stimulate contractions. NSAIDs are known to cause stomach ulcers by the same mechanism, just in reverse. We have other drugs that mimic other eicosanoids, and thus it is clear that this group of hormones is critically important pharmacologically, but also physiologically. PGI2 is an important vasodilator in the arteries and also inhibits platelet aggregation. Imagine the effect of NSAIDs on this. Indeed, we do see some risk of cardiac-related events with NSAIDs for this reason. The key is this. How does your body know which prostaglandin to make? The fatty acid it selects from the membrane may have a role, but it importantly is often the stimulus for the release in the first place. Is the cell infected? Is there damage? Is it time to renew the cell cycle? These stimuli and environmental conditions are likely more important for deciding on the prostaglandin selected than the fatty acid composition of the membrane, i.e. the amount of dietary omega-3 versus 6. Leukotrienes are very similar eicosanoids, we just know less about them and know fewer of their functions. They appear to exist in a group of pro-inflammatory molecules which seem to stimulate the immune system by their effects. Notice that leukotriene B4 and B5 directly stimulate neutrophil chemotaxis or transport to the cell. Instead of COX, these are metabolized through 5-LOX or 5-lipoxygenase, and we also have pharmacologic agents to prevent this and LOX inhibitors from a drug called xylitol, or even direct leukotriene receptor antagonists like montelukast. Ultimately, these drugs are mostly only useful minorly for allergic rhinitis or asthma, but there could be undiscovered use cases for drugs like this in the future. An incredibly important eicosanoid specifically is thromboxane A2. Created through COX, TXA2 has critical function in primary hemostasis. That is, damage to the endothelium, for example trauma, leads to exposure of a protein on the surface called von Willebrand factor. This attracts and binds circulating platelets to begin primary hemostasis. After binding, platelets are activated, where they release ADP and calcium from their contained granules, but also this thromboxane A2, which binds to TXA2 receptors on the platelets, which further activates more platelets and creates this platelet activating feed-forward loop necessary for effective and adequate platelet aggregation. These effects are counterbalanced by PGI2, which, remember, decreases platelet aggregation and causes vasodilation. In this way, NSAIDs could also theoretically lead to delayed wound healing and excessive bleeding, but it is through this mechanism by which aspirin, or ASA, is suspected to protect patients who are having a heart attack and to prevent them from developing an MI by inhibiting the platelet aggregation cascade. We can only clot like this using our eicosanoids. Now we introduced that the metabolites in eicosanoids made by omega-3 and 6 fatty acids can contribute to both pro-inflammatory and anti-inflammatory action. But don't we only want anti-inflammatory action? Isn't all inflammation bad? Well, this is unfortunately a common misconception. Inflammation is in fact a highly evolved infection protection mechanism that all living things possess to defend ourselves from invaders, locally from our own microbiome, but also exogenously. Not only that, but our immune system can also defend against cancer development. The dark side of inflammation, of course, is that it causes a lot of collateral damage, and if left unchecked to continue for too long, can cause irreversible and irreparable damage. The key is balance, being able to generate an immune response when we want to, but then controlling it. Lack of immunity leads to more frequent infections, but also strange and atypical infections. For example, take progressive multifocal leukoencephalopathy, a brain disease caused by a virus called John Cunningham virus that all of us possess, but only starts to cause problems when we have virtually no immune system to control it. But excess immune reactions or autoimmunity can also cause frank disease, for example, IBD or type 1 diabetes. The question is whether or not the choice between our dietary polyunsaturated fatty acids could lead to some regulation of the balance of this process. Here's what we do know. We can, in fact, at will, change the plasma membrane composition of our cell membranes. By eating more omega-3 fatty acids, more will appear to reside in our plasma membranes on a biopsy specimen with lipid raft analysis. Here's what we also know. Enzymes that are important in the inflammatory cascade, for example, complicated cell regulators like peroxisome proliferator activated receptor or PPAR alpha and gamma are differentially stimulated by different prostaglandins. If it's true that different PUFAs generate different eicosanoids, a plausible mechanism is in place. What we are missing is if our body preferably chooses different PUFAs from our membrane via PLA2, if they are over or underrepresented, or if that does not matter at all. Ultimately, this is an interesting concept, but at the moment is hypothesis generating at best. Further, the notion that certain prostaglandins and by proxy their eicosanoid precursors are strictly inflammatory or strictly anti-inflammatory is a gross oversimplification. Suffice to say, we already know that, for example, EPA and DHA metabolites can act as both pro-inflammatory and anti-inflammatory mediators. Some argue they are less pro-inflammatory, but now we begin to split hairs, and the ultimatum is that we need to see some form of clinically undeniable difference between patients with plasma membrane incongruence or a Mendelian randomization study describing phenotypic differences based on these compositions to know the truth. To throw a final wrench in the thinking, there is an abundance of eicosanoid subtypes that are produced by these fatty acids we just simply don't understand. For example, some molecules like resolvin E1, D1, and protectin D1 are all anti-inflammatory and might be derivable from both omega-3s and 6s. Understanding the complex interplay is beyond the scope here, but is worth introducing in case you are considering diet manipulation on these mechanisms. As a clinical aside, we of course stop this process entirely with corticosteroids, which inhibit PLA2 and thus stop inflammation from this perspective. At the end of the day, we want our patients in true center of this spectrum, not slightly towards being immunodeficient and not slightly over-inflammatory. The concept that a patient could be slightly over-inflammatory is tenuous. This presumably is the hypothesis of those who emphasize strongly that everyone needs to take omega-3 fatty acids as supplements. We already know that there are some plausible mechanistic benefits of why omega-3 supplementation may be beneficial, specifically for VLDL and triglycerides, but in patients who don't have this problem or who are managing it via a variety of other means, including diet, pharmacology, exercise, and other means, the argument that they are simply suggestible to all comers may be premature. Think about the impact of the omega-3-6 PUFA ratio in the form of a pie chart. Really what we care about is if there's one, some sort of actual disease prevention benefit in skewing this ratio, and two, if there is some sort of subjective quality of life or longevity benefit to changing the ratio. Being scientifically honest, how many unknowns are playing a part in that pie chart? I.e. the incredibly complex impact of genetics, epigenetics, overall non-PUFA diets, the dietary zero-sum game, physical activity, sociocultural phenomena, to name a few. In this way, we should contextualize the possible effect that this ratio could have on these factors. It's possible that it has some role, but in reality, it is simply too confounded by other possible inter-individual or inter-population differences. It's difficult to pin the difference on this ratio at the moment when there's simply so much that is unknown about the pro-inflammatory and anti-inflammatory features of each metabolism pathway. A reminder that inflammation is one biological process that is a factor in disease and cell death, amongst many others. So perseverating on inflammation at this level and at this context is likely to lead to missed emphasis on potentially more impactful mediators of the positive outcomes we've listed above. We still don't know much about these biological interactions, but for the plausible cardiovascular benefits via reduction of VLDL and reduced triglycerides comparatively to a high saturated fat and carbohydrate diet, inclusion in the diet is justified on that basis, with inflammatory benefits being largely hypothetical. As usual, the well-rounded diet is advisable. Now last, but certainly not least, is the use of phospholipids from fatty acids of all types, now as intracellular or extracellular second messengers, meaning that they play a role as a part of a larger enzymatic and cell signaling cascade, like in G protein coupled receptor pathways. Fatty acid molecules like phosphatidylinositol biphosphate, or PIP2, can be cleaved from the plasma membrane just like AA or EPA, yielding second messengers like inositol triphosphate, or IP3, and now a free diacylglycerol, or a DAG. IP3 can stimulate calcium release directly from the endoplasmic reticulum, and both calcium and DAG can stimulate an important protein called protein kinase C, which phosphorylates a target protein for an effector downstream enzymatic effect involved in many very important cell-regulating behavior. Examples of these pathways include fatty acids as cell signaling molecules, include critical receptors like alpha-1 adrenergic receptors, histamine receptors, and serotonin receptors. DAGs, importantly, specifically with relationship to overabundance in the plasma membrane with their hydrophilic head group facing the cytoplasm, may be at least part of the cascade of problems with overstimulation of PKC with regards to insulin resistance in the muscle, which culminates in less GLUT4 receptor translocation and then less insulin sensitivity in the muscle.